And obviously, there is going to be developmental earlier and earlier stage when transferred to the pedigree, like if you pass through the pedigree, your disease will come earlier and earlier. The number of repeats will start increasing. So, actually, very originally, the fellow from, I should remember his name, he's from Harvard. He is the one who went to this particular family. This is another family which moves around from Venezuela in Lake, Maracaibo. And this particular lake, people live on it and they keep moving around. But unfortunately, for doing genetics, we need a lot of pedigrees and collection of a lot of DNA samples for each individual. So once you get them and do an experiment, and then the board is missing, you cannot even repeat the experiment. So it was really a tremendous task for him to clone this particular Huntington gene. It took a while. Finally, they got it done. So it's a large pedigree, but introduced by only one individual. So one individual had a mutation to begin with, and the entire lake population was loaded with this Huntington disease. Okay? Fragile X. This is another expansion disorder. It is associated with mental retardation. It has got the gene, which is the Fragile X mental retardation gene, FMR1. I told you my postdoc is the one who cloned this gene. Subsequently, after he left my lab, and sometimes we see the hemophilia, but he cloned this gene subsequently. And sometimes pre-mutations can develop into adult onset, neurological disorder, and obviously Fragile X is associated with all sorts of tremor and other associated events. So that's the way a person with Fragile X syndrome look like. And the X chromosome has got a site which looks as if it is discontinuous, and that is called a fragile site. That's what the individuals have. So cytogenetics will first detect it very easily. But look at this. There is one more thing called a pre-mutation. Frequency of expansion of a pre-mutation. Like, for example, if there are a number of repeats, and 56 to 59, the frequency expansion of full mutation, which is disease-causing situation, is 14%. Whereas if the number is tremendously high, like 100 repeats are there, your disease-causing is going to be essentially 100%. So the number of repeats really matter for this Fragile X as well. Okay? But this is not present in coding sequence, which is present in 5' UTR. So carriers of pre-mutations, premature ovarian failure by age 44, and they have got myotonic dystrophy. So these are the things that you should know about carriers of premature ovarian failure by age 40. And then there are other things called myotonic dystrophy, where this is an autosomal dominant myopathy, myotonia, and wide variety of other issues are there with that disorder. And pre-mutations are asymptomatic in this case. Okay? This is myotonic dystrophy. This is also an expansion disorder. So if you look at this person, and if you look at these eyes are really a hallmark for giving out the myotonic, because they have the tone of the muscle will be altered in this myotonic dystrophy. Okay? The weakness of muscle symptoms. And there are a wide variety of other disorders which are also involved in this repeat expansion, Friedreich's ataxia, spinocerebellar ataxia, etc. So now we have covered many disorders with respect to repeat expansion. So you should be able to at least remember the classical ones, like Huntington, Fragile X, myotonic dystrophy. These are the three things that you should remember. The rest of them I don't care. Okay? So now we have got maternal inheritance disorders. Okay? It's very remarkable that in the entire world, in the culture, we always believe in paternal inheritance. Mostly the males are given some kind of priority. I mean, it's not really a gender situation here. But there is one religion, Jewish religion, that is not the case. If mother is a Jew, it doesn't matter who the father is. The next person is a Jew. Yeah. So it's a maternal inheritance. It's the only religion which believes in maternal inheritance. That's very remarkable because they found essentially maternal inheritance because of mother's mitochondria. So the mitochondrial genome is approximately 16.5 kilobases. That has got several genes. It has got 37 genes which are involved in oxidative phosphorylation, there are a variety of ribosomal RNAs, there are 22 transfer RNAs. That means the mitochondria have got microribosomes. It can code for its own proteins, it can synthesize its own proteins, it has got all the machinery for synthesis of its own. So the mitochondria makes, therefore there are 22 transfer RNAs. So if there is a problem with any one of these proteins, obviously you will have problems in oxidative phosphorylation. So you've got problems in respiration, etc. Therefore, even in yeast, long time ago, they isolated petite mutants which are defective in respiration. Okay? And because of this, you have wide variety of other possible side effects, and therefore you will have mitochondriopathy because of the problems associated with these gene mutations, particularly transfer RNA mutations. It's very simple. If alanine transfer RNA is mutated, it is not going to put the alanine back in proper places, and therefore you will have tremendous defects in oxidative phosphorylation, etc. So there are almost greater than 100 mutations have been reported. But if you take a look at how the mitochondrial inheritance is going to be purely maternal, sperm is loaded with mitochondria also. But the sperm has to swim to reach the egg. Therefore, it loses all the mitochondria. Energy is totally lost. Maybe it will have 1 or 2 mitochondria. But the ovum is the one which carries all the mitochondria coming from the mother. Therefore, when it fertilizes, what you carry is maternal mitochondria, and paternal mitochondria are diluted. There is 1 or 2. We're not telling that there is no paternal mitochondria at all, but 99% of the mitochondria are derived from the mother. So now, if a replication occurs, the mitochondrial genome has to also replicate by itself. It's a genome, after all, it's a mitochondrial genome. So let us say that these are all the mitochondria, but there is a mutation in one particular, 1 or 2 mitochondria. One genome has been mutated. Now what happens is, when the mitochondria are really replicating, and it all depends on your cytoplasmic segregation, depending upon how much of a cytoplasm and how these mitochondria are distributed in cytoplasm. Suppose if one particular cell gets these mitochondria which are mutated and the other one gets a limited number, still mutations are there, but this is a limited number, and therefore the other normal mitochondria dominate, and therefore they will give you a normal phenotype. But if you get this kind of a situation, you call it a disease phenotype because of mitochondria. Or therefore, this kind of a situation is called homoplasmy, one of them. That is, one daughter cell may receive only pure population of mutant mitochondria. That is homoplasmy. Whereas heteroplasmy means it is going to get both mixtures in different populations, and therefore heteroplasmy will not lead to the disease, whereas homoplasmy of the mitochondria will lead to the disease. Okay? So, as I just now pointed out, there is no mutant mitochondria from male, because even, let us say there is a mutation even in the mitochondria, it's going to be 1 or 2, right? And therefore, when it splits, it's insignificant, and it's not going to be really problematic. So, male will not transmit any mutations, even though he may carry the mutations derived from the mother before, but he will never transmit the next generation. There is one disorder which is associated with the Leber hereditary optic neuropathy. So initially, mitochondria reduced before amplified in mature oocytes, and this particular disorder is definitely due to the mitochondria. So if you take a look at here, that's a male and a female. The female transmits the disease. So if all children got it here, but only this particular one will not carry it because the male mitochondria, even though he's a mutation, 1 or 2 mitochondria will be transmitted. That is not going to express the disease. Whereas in the female, tremendous amount of mitochondria, therefore the children will definitely get that. Okay? So that is the maternal inheritance. So if you take a look at this kind of a pattern, somebody gives you the pedigree like this. Okay? And somebody puts side by side some other different inheritance like recessive, pseudo, autosomal, etc. You should be able to figure out it is purely maternal because there's nobody else is coming up. Okay? So by looking at the pedigree, you should be able to tell it is mitochondrial inheritance and maternal inheritance. Okay? So you now learned several types of pedigrees, and all the pedigrees are about 7 or 8 types of pedigrees you learned. Okay? So each pedigree, we may be able to test you in one of these things. So just I will give you one of these kind of a diagrams and ask you to determine whether it is maternal, autosomal recessive, dominant, or male predominant or female predominant and things like that. Okay? So that would be, this chapter will cover you. Those are the pedigrees that you learned. So I hope now you know. Previously I asked you, have you drawn family pedigrees? You said yes. Now I'm sure you know more pedigrees than what you learned before. Okay? So far, we learned about gene deletions, gene inversions, rearrangements, a little bit of mutations, and mostly we're all thinking about single gene disorders. When it comes to more than one gene mutation, let us say you have 10 genes which are causing a disease. We're talking about all these 10 genes are located at multiple loci. So now, if you apply Mendelian independent assortment and segregation, etc., it is very difficult for you to predict if a parent carries 10 genes mutations independently, and then a mother may carry some other kind of a disorder, we don't know. Let us say these are all multifactorial. So when the progeny is there, because of independent assortment, you will not get all the 10 to the next progeny. And therefore, on the other hand, subsequent generation, it may be affected because, again, different combinations you may get that. So multifactorial inheritance is not trivial to understand, not trivial to do genetics on it. It's extremely difficult. So that is the reason why almost quite a bit of the disorders which are multifactorial, such as diabetes, hypertension, lots of disorders are there, Alzheimer's and things of that kind, their genes have not been identified yet. Maybe one or two would have been identified, but the entire complement of the entire genes which are causing these disorders are not known. Okay? So this is all meant for the future research programs. So there are a lot more multifactorial disorders for which the molecular basis is really rather unknown. So that is all for your generation to do future research on the multifactorial genomes. We have done our job, that's all meant for you to do. Okay? So one of the ways that you could at least handle these disorders is by looking at studies such as familial clustering. Okay? Only within the family that you can study these. You cannot just take different families and study them because, as I told you, inheritance is complex. So if you have a single family, same gene-to-gene interactions and gene-environmental interactions are possible. See, on the top of these genes, you have also environmental effects are there. For example, you may just simply get exposed to the sunlight. DNA repair genes will go bad. We don't know. Or simply you may take a food in which they are loaded with some kind of estrogens or whatever. So steroids. So you may get gene expressions will be affected. So these are all environmental factors that could be, like anything that you eat food, for example. Okay? So all those things will also expose the gene-gene interactions to alter. And there are also what are called modifier genes. That is, the genes which are controlling the transcriptional levels of other genes. Those are modifiers. Or, as you already learned about histone code, you learned in the previous chapters. So that is a modification. So that's a modifier gene. So depending upon the modifications, also complexity will arise. So the only way that you could study multifactorial inheritance is by empirically derived risk figures. Otherwise, it is pretty hard.